For today. We have Nishtha Bhargava from CSIR Institute of Genomics and Integrative Biology, India. So you can share your screen and yeah, we'll get the session started.
Okay. I'm audible and the screen is visible?
Yeah.
Yeah.
Just a second. Okay. Hello, everyone. I'm Nishtha from India. I'm a PhD student in CSIR Institute of Genomics and Integrative Biology in Delhi. Today, I'll tell you about the fact that heat shock induces alterations in small extracellular vesicle protein and lipid cargo. These observations were obtained when we were trying to understand the extracellular effects of disruption in protein homeostasis or proteostasis. My slides do not seem to be moving. Okay. Protein homeostasis or proteostasis is an intricate mechanism within cells which maintains functional proteins, their basic native conformation, and this is important for cellular health to be maintained. In perspective of metazoans or multicellular organisms, when we talk about things like genetic mutations and environmental factors that are constantly challenging proteostasis, in metazoans, the different kinds of cell types that are there, they are not equally capable of mounting the responses that will maintain proteostasis, which means some cells could cope up with the environmental stresses, whereas others may not be able to. And this poses a threat to the organism health. But since this does not happen in real life, it points to the fact that there is some sort of cell-to-cell communication that is happening, maintaining proteostasis at the organism level. And small extracellular vesicles are one kind of mediators of cell-to-cell interaction and very important mediators as such. And indeed, chaperones that are helping molecules, part of the proteostasis network, they are known to be transmitted through EVs. And EVs can also give resilience to recipient cells in case of heat shock. But in neurodegenerative disorders, in which one of the hallmarks is aggregation of proteins, there the role of EVs is somewhat contradictory. In some studies, the role of EVs is beneficial, and in other, it's detrimental. So we are not, all the mechanism underlying the role of SEVs in proteotoxic stress as such is very poorly understood as such. And especially the cargo that is carried in small extracellular vesicles during proteotoxic stress and its transmission between cells is even further poorly understood. So we wanted to embark upon this question. Study design was pretty straightforward. We took cultured cells, we took HEC293 T cells and wanted to isolate EVs from them in control versus heat shock conditions and see what kind of protein and lipid cargo they contain. First of all, we know that the timing of isolation of the EVs can play would be a major property that would affect the EV cargo. So we wanted to see at exactly what time point the HEC293T cells will elicit the heat shock response, which is basically the upregulation of molecular chaperones HSP70 and 90. And we saw that after heat shock at 42 degrees for 30 minutes, when we gave them recovery at 37 degrees, the increase in HSP70 and 90 happened across around 4 hours, and then it went back to control levels. So this 4-hour recovery time point after heat shock became the time point where we will isolate our EVs. We isolated small extracellular vesicles by size exclusion chromatography because we wanted them to be as pure as possible. When we plotted the particle concentration versus protein concentration, we could figure out in which fractions the particle concentration is high, whereas the protein concentration is low, because the free protein will elute at the end. And then these fractions were the ones we characterized for EV size, morphology, and also the markers. So as you can see, the size range was within the expected range, as also the morphology can be seen from the TEM image here. And when we immunoblotted for various SEV markers such as syntenin, TSG11, and CD81, the EV isolates were positive for that and negative for the EV marker calnexin, which shows that we had pure EVs. Once we had these for both the control cells at 37 degrees and the cells that had experienced heat shock, we went forth and looked at what kind of protein cargo these contained. So first, if we look at the PCA plot, we can see that the control EV proteins and the heat shock EV proteins are separated out on a principal component analysis, showing that there are differences indeed in these two sample types. And when we looked at the protein identities, there were around 20 odd proteins which were differential between the two kinds of EVs, the control EVs versus the heat shock EVs. And when we applied the cutoffs of fold change of plus or minus 2 and a p-value of less than 0.05 after correction, we came down to 5 proteins which were differentially present in the two kinds of proteins, two kinds of EVs, sorry. The 4 proteins were enriched in heat shock derived EVs, whereas apolipoprotein B100 was depleted. Interestingly, we didn't find any of the canonical molecular chaperones like HSP 70, 40, or 90. However, this particular protein, vitronectin, is recently discovered to be an extracellular chaperone present in human serum, and it can prevent protein aggregation in vitro, both fibrillar and amorphous kind of aggregation. And apolipoproteins are implicated in diseases like cerebral palsy and Alzheimer's. And recently, their role in proteostasis is also being uncovered. So heat shock EV protein cargo is definitely altered compared to control EVs. And however, the mass spectrometer that was available to us had lower resolution. And so if we run this kind of proteomic analysis on a more sensitive mass spec, this could potentially lead to several more revelation of further more changes under proteotoxic stress. Moving on to the lipid cargo under heat stress, we identified 600 to 700 lipid species in each of the samples, heat shock EVs as well as control. And out of them, around half of them, around 230 of them were differential, either increased or enriched or decreased in the heat shock EVs. Now, this is a list of the species of lipids that we detected. I'll not go into them, detail of them for paucity of time, but specific interest is in ceramides and sphingomyelins because they are known to be involved in proteostasis or protein stress related changes. I'll just briefly introduce you to them. Ceramide and sphingomyelin both are present, are formed in the ER and can be converted from one to the other. Interestingly, exogenous addition of ceramide or modulation of ceramide endogenously to lead to an increase actually upregulates small heat shock proteins, which are an integral component of the proteostasis network. Another study tells us that neutral sphingomyelinase 1 leads to an increase in ceramide inside the cell, which further leads to heat-induced apoptosis. And to refresh your memory, neutral sphingomyelinase 1 is an enzyme that is involved in the production of EVs through the non-escort pathway. And lastly, there are studies which show that during the effect of antidepressants, ceramides in endoplasmic reticulum are increased, which further leads to stress inside the brain cells. So all in all, we see that heat shock EV lipid cargo does indicate alterations that compared to the control EVs, and the lipidomic analysis of more biological replicates would be required to have a deeper understanding of how these lipid species changes are actually relevant in the case of heat shock or any proteotoxic stress for that matter. And this study is on as we speak. And to conclude, I would like to say that heat shock does cause considerable changes in the cargo of EVs, and some cargos are there that have known functions in proteostasis, but there are a lot many species that have their potential role has not yet even been uncovered. And this study thus underscores the significance of exploring cargo of SEVs with the aim to understand the mechanism in proteotoxic stresses. And here are a few open questions, and I'm open to your questions and suggestions. Thank you.
Thank you, Nishtha. So today, since we're running a little short of time, we don't have the time to actually ask the questions, but all of the posters are available on the Discord link. So whoever has any questions,